Okay, so MPS IIIB is mucopolysaccharidosis 3B. So it's one of a family of disorders where there's an abnormal accumulation of mucopolysaccharides in the tissues. MPS IIIB is a disease where there is systemic involvement in the disease, but there's also a prominent involvement of the brain, which leads to slowing of mental development with cognitive decline, typically starting around about 4 years of age. It's a devastating disease, and historically there has been no treatment for patients with this disease. So SBC-103 is a recombinant form of the human natural enzyme that's been developed as an enzyme replacement therapy for patients with MPS IIIB. MPS IIIB has actually been a disease where there were some concerns that it may not be possible to develop an enzyme because attempts to make the enzyme about 10 years ago using cell culture, which is the normal method for making these enzymes, were unsuccessful. So we were able to successfully make the enzyme with the right characteristics that allowed uptake into the key cells and tissues. And then at this meeting last year, we presented data that showed that if you gave this enzyme intravenously, you were able to reduce the abnormal material that was accumulating in the liver, but also importantly in the brain, where many of the complications occur. And we're very excited now. We've actually just started our first human study in patients with MPS IIIB with SBC-103. We have a presentation at this meeting, and that presentation is looking at some aspects of the disease in detail in some untreated patients using some state-of-the-art methodologies. So one of the things that we've been looking at is there was some data about 2 years ago that was published in a mouse model that showed that the barrier between the blood and the brain in these patients, in these mice, looked abnormal due to a large amount of accumulation of the abnormal substrate in the blood vessels supplying the brain. This hadn't been previously looked at in humans, and we did a study looking at patients with the disease using a state-of-the-art method for looking at the integrity of the blood-brain barrier, and we're presenting that data at this meeting. The bottom line is there is some evidence of a subtle defect of the blood-brain barrier in patients, but it's not a major defect of the blood-brain barrier. And that study actually provides a very important foundation of information that we will be building on as we move forward with SBC-103 in the clinic.